everyone. Thank you all for joining us for our February Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking everyone who supports the HDF. It is because of you that we're able to fund the exciting science that's transforming what we know about Huntington's disease. And a big thanks to all of you for tuning in. Thank you for being here. I'm happy to introduce our speakers today who will be sharing their work on something that is really important in Huntington's disease research, and that is the repeating CAG genetic letters that lead to HD. We've known for over 30 years that this is the cause of HD, but more recently, we've learned that there are small changes within the CAG repeat that can have a big impact on when someone develops HD, like a 10-year difference. So clearly there's more to this story, which our speakers will tell us about today. We are joined today by Dr. Chris Kay and Dr. Jong-Min Lee. Dr. Kay is a geneticist and molecular biologist at the University of British Columbia in Vancouver, Canada. He studies genetic modifiers of Huntington's disease for discovery and understanding of new therapeutic targets. His research also focuses on the significance of somatic CAG repeat instability in HD and how the gene that causes HD varies between people around the world. Dr. Lee is an associate professor at the Massachusetts General Hospital and Harvard Medical School, specializing in human genetics with a focus on identifying genetic modifiers of Huntington's disease. He's a member of the GeM-HD Consortium, and his team has uncovered that DNA repair genes are involved in somatic CAG repeat instability, and they play a crucial role in modifying the age of onset and other key features associated with Huntington's disease. Currently, his laboratory is translating these genetic insights into novel treatment strategies for Huntington's disease. Thank you both so much for joining us today to share your work. Before I pass it over to Chris, I want to remind all of you that you can ask questions in the Q&A box throughout the talk. So try to use the Q&A box, not the chat box. We should have about 20 minutes at the end for Q&A. So send your questions in now as you think of them so that you don't forget, and we will address as many at the end as we can. Also, a note that this will be recorded and available on our website and YouTube channel in about a week or so. So if you want to watch it again or share it with someone, it will be available for you to do that. And again, thank you all for being here, and I will pass it over to Chris.
All right. So thank you very much, Sarah, for that kind introduction. I'm based at the University of British Columbia, where I work on Huntington's disease and have worked on HD for quite a few years now. But I'd like to start off the presentation by clarifying something, which is that even though I am Canadian, I am not from Canada. Everyone assumes that I am from Canada. I'm actually from California and Florida. So I was born in the Bay Area and then moved when I was younger to Florida. And then moved back to California to go to college in the LA area. And then after doing college in LA, I actually went back to Florida again to complete university training in genetics. And I think this is sort of a funny relationship that I have with California and Florida, given these have sort of become arch nemesis states in the United States. And so perhaps because of the political situation, I said, you know what? I'm going to have a change of setting and move to Vancouver, Canada. Vancouver is in British Columbia, and Canadians sort of like to think of British Columbia as the California of Canada, which at least in the summertime is true. So we have beautiful mountains, beautiful beaches, a mild climate compared to the rest of Canada. But do not be fooled. Most of the year, it actually looks like this. It's a very rainy place. So while it might be the most mild place in Canada, it definitely is not the same weather as California and Florida. However, it was in Vancouver that I started working on Huntington disease, where I joined the research group of renowned HD researcher, Dr. Michael Hayden, who I still work with today. Michael Hayden set up the Center for Molecular Medicine and Therapeutics, which is a dedicated research institute at the BC Children's Hospital, which you can see in the middle panel here. We don't have this beautiful view of the city, but it's just a short bus ride away. It's really quite a remarkable place to work. And many of the key discoveries in HD genetics were actually done right here in Michael Hayden's group in Vancouver, which is how I started working on the genetics of Huntington's disease. And what I'll be speaking to you about today is about some of what we know about the CAG repeat and how that knowledge has actually changed a bit in just the past year or two. So I think as many of you watching are aware, a person who has Huntington's disease has two CAG repeats. So they have a shorter CAG repeat, which you would call the normal CAG repeat, and they have a longer CAG repeat that we would call their Huntington disease CAG repeat. So the normal CAG repeat is typically below 35 CAG repeats. And the repeat that causes Huntington's disease is one of two categories. It's either a reduced penetrance allele, which would be between 36 and 39, which means you may develop HD, or it could be 40 and above, which normally is reported as saying you will develop HD. What's interesting is that both of these CAG repeats, uh, they form this translated product, this protein product called a polyglutamine, because the CAG repeat actually encodes this protein building block called glutamine, which is represented actually by us biologists as a Q. So however many repeats you have on your normal CAG create a corresponding number of glutamines, and however many CAG repeats you have on your HD CAG repeat cause a longer number of glutamines. And so we call this the Huntington disease polyglutamine. And it has really been a dogma in the field for, over 30 years, that it is this Huntington's disease polyglutamine that is the most likely toxic causative agent of Huntington disease. So what does the CAG repeat look like? I think people are familiar. It's this repeated CAG, but it's important to know that the CAG repeat, it occurs within a surrounding DNA sequence. So as you can see in this slide here, there's a sequence of nucleotides, these AGCTs that occur before the CAG repeat, as well as after the CAG repeat. So there's this conserved sequence with an underlined section here that normally occurs right after the CAG repeat. And then actually what a lot of people don't know is that there's another repeat that doesn't really change much in people called the CCG repeat that's just adjacent to the CAG repeat. So all that happens when you have a Huntington's disease CAG repeat is that CAG segment becomes longer. So the segments around it stay the same, but the CAG repeat becomes longer. Um, so work that I did a number of years ago actually characterized how the CAG repeat varies between people, because even among people who don't have Huntington's disease, the CAG repeat can be quite different between people. So everyone who's on this webinar right now has a different number of CAG repeats in their genome. It just happens that people who have Huntington's disease have one CAG repeat that's very long. And we estimated at the time that roughly about 1 in 500 people have a CAG repeat of 36 to 39 in the reduced penetrance range. And roughly 1 in 2,500 people have an HD repeat above 39, so 40 and above. And it was really striking at the time that we found these numbers that this is a bit larger than the number of people who are actually diagnosed with Huntington's disease. So those repeats can occur actually with some frequency in the population. So what's been a real focus of HD research in the past year or two is actually a return to something that we knew many years earlier, which is that the number of CAG repeats not only varies between people, but it can actually vary between different cells of your body. And we know that there are specific cells, specific tissues of your body, where CAG repeats tend to expand further in people who have HD. So we knew a long time ago that it was actually specific regions of your brain that are affected in Huntington's that actually have the most of these extra CAG expansions in a person who has HD. And we also knew for some time that in sperm, there's also a propensity for longer CAG repeats, which is why people who inherit the repeat from their fathers have a higher likelihood of getting a longer number of CAG repeats. So it's been known for some time that there are specific cells in your body where the CAG repeat will expand further. Um, Really exciting work that has come out in the past year or two, and this has been enabled by a lot of technological advances, has shown in detail that there are specific types of neurons that have very large expansions. On the right here are a bunch of different types of neurons. This is a study from Steve McCarroll's group at the Broad Institute in Boston. And what they did is they looked at how long is the CAG repeat in different types of neurons from brains of people who had HD. And they found that these medium spiny neurons, which are abbreviated as MSNs, they tend to have repeats that are not only more expanded than other neuron types, but some of them have many, many CAG repeats, in some cases, hundreds of CAG repeats. And so that's led to this hypothesis in the field that perhaps it's these large CAG repeats in these vulnerable neurons, in these medium spiny neurons, that could be leading them to be vulnerable in Huntington's disease. Because it's quite a coincidence that these neurons are the ones that tend to die in Huntington's disease, and they also have these very long, highly expanded CAG repeats. Another reason why we care about the number of CAG repeats is that we've also known for some time that the number of CAG repeats that you inherit, the number of CAG repeats that you get from a test result, it associates with when you tend to develop HD. So this is a simple plot developed from clinical data from many, many people who have HD that shows the relationship between the number of CAG repeats that you have in a test versus when, on average, you tend to develop the first symptoms of Huntington's disease. So, for example, someone who has 43 CAG repeats, on average, they would tend to present at about 48 years of age. However, we do know that there are people who present earlier than expected based on their number of CAG repeats, and people who present later than expected based on their number of CAG repeats. And this has allowed us to search for potential genetic factors in these people as to why they may be presenting Huntington's disease earlier or later than you would expect based on how many CAG repeats they inherit, how many CAG repeats they test for. And Jong-Min will speak a bit more about this in his presentation. But I do want to talk about how we sort of have categorized these modifiers that cause people to present HD earlier or later. And the way that I like to think about it is that there are genetic variants, modifiers, that occur in the Huntington's disease gene, and they tend to occur in that CAG and CCG repeat region I mentioned earlier, as opposed to modifiers that occur elsewhere in your genome in other genes. And these modifiers that are in the gene sometimes can have a profound effect by about 5 or 15 years in terms of earlier or later presentation of Huntington's, whereas these modifiers in other genes elsewhere in your genome have a somewhat smaller effect of around 1 to at most about 5 years difference of onset. So the reason I'm bringing up these modifiers is that when these modifiers in other genes were discovered, and Jong-Min was instrumental with the GeM-HD Consortium in some of this work, it was identified that some of these genes, which are the ones highlighted in red here, belong to this specific pathway called DNA mismatch repair. And it was understood at the time, and now in a lot more detail, that these mismatch repair proteins actually impact CAG repeat instability and expansions in not only in cells and mice, but potentially also in people. So it shows that these modifiers can impact Huntington's disease onset and may potentially do that by impacting somatic instability. So let's return to these modifiers that are in the Huntington's gene. And this is work that I've been involved in for many years. So as I mentioned, these modifiers can change the presentation of HD by many years. It's a profound impact when someone has a variant in this region. And so you might recall from what I said earlier that you have the CAG repeat, and then you have that CCG repeat that's next to it. Well, so the sequence in most people looks like this. So you have a CAG repeat, and then the second to last set of letters is actually what we call an interruption. So rather than a CAG, it's a CAA. And the second group of letters in the CCG repeat is a CAA. So what happens in these individuals who have these variants in the gene is that they lose these interruptions. And so rather than having those gray sequences, they actually have a pure uninterrupted CAG repeat followed by a pure uninterrupted CCG repeat. And we call this either the CAG loss of interruption or the CCG loss of interruption. And so that we don't have to say that over and over again, we will call that the CAG-CCG loss of interruption. And the impact of these variants is, as I mentioned, quite profound. So if you lose both of those interruptions, you will present Huntington's disease on average about 12 and a half years early. If you lose just the CAG interruption, you present in our data about 7 years early on average. And then if you lose just the interruption in the CCG repeat, remarkably, you also have a much earlier presentation on average. And brilliant work by my colleague Jessica Dawson has also shown that it is the progression of Huntington's disease that is also accelerated when people have these loss of interruption variants. So given what we know about the presence of somatic instability in Huntington's disease in the brain and how longer repeats cause earlier onset, a natural question would be, do these loss of interruption variants cause more CAG repeat expansions? And this is work we've done here in Vancouver for the past year or two. The surprising finding is that they do not increase somatic instability. So we first looked at blood, because blood DNA, of course, is very easy to access from patients. This is work that was led in many ways by my colleague, Haley Finlay Black. And what you see in this plot is as the number of CAG repeats go up, instability goes up. So in blue, this is people who have the canonical sequence. They have the interruptions as normal. But people who have the loss of interruption, which are depicted in red, although they have more instability with increasing number of CAG repeats, they don't have higher instability than people with the ordinary sequence. So whatever's happening in individuals who have this loss of interruption variant, it's not dramatically increasing somatic instability in their blood. So you could argue, well, perhaps there's something happening in the brain that's not happening in the blood. Well, we've looked at that too. And the story, unfortunately, in terms of understanding this, is the same, that there's no increase in somatic instability in different regions of the brain in people who have these loss of interruption variants. And in fact, we have some suggestive evidence that instability may actually be lower in people who have the loss of interruption variants. So if the loss of interruption in the CAG and CCG repeats doesn't increase expansion, well, how does it accelerate Huntington's disease? And the answer is that we don't know. We have many hypotheses that we're testing right now, but it seems to be occurring through some different pathway or mechanism than somatic instability. So I want to return briefly to those very long repeats that I mentioned earlier that we now know occur in specific types of neurons that are affected in Huntington's disease. Because perhaps we're missing these long repeats in people who have the loss of interruption variant. You know, we don't really know a lot about these very long repeats, when they occur, what is their relationship to Huntington's disease. And so what we've done here in Vancouver is we've started looking at these very long repeats in brain tissues from people who have different stages of Huntington's disease. And this has been enabled by really visionary work by Michael Hayden and Blair Levitt here at UBC, building the UBC HD Biobank, where we have now collected through relationships with patients and outreach caregivers, as well as genetic counselors, and through the Huntington Society of Canada, a streamlined brain donation process here in Canada, where we now have about 250 Huntington's disease brain tissues that have been graciously donated. And when we look at these brain tissues, so we've looked at a few different regions that are affected in Huntington's disease. We find that in later stage Huntington's disease, there are these long repeats beyond 150 CAG repeats that occur. However, when we look at tissues of people who haven't had HD for very long, but do have symptoms, we haven't seen them so far. So, for example, this is an individual who, here in Canada, in British Columbia, we have medical assistance in dying. And so this individual had onset of Huntington's disease at 72 years of age. And chose to do medically assisted dying approximately 1 year after they developed chorea, psychiatric symptoms, all the typical symptoms of Huntington's disease. And when we looked at the presence of these very long repeats in brain tissues from this donor, we didn't see any. So it leaves wide open the question as to what is the significance of these long repeats in earlier stages of Huntington's disease. So what we're doing right now in Vancouver is looking at the presence of these long repeats in different kinds of neurons from brain tissues. And we're hoping that this will shed light on some of the questions that we now have about the impact of these expansions on how Huntington's disease develops. So I need to wrap up and hand it over to Jong-Min. What is the impact of some of these studies with respect to therapeutics? There's actually quite a few considerations and impacts. So the first is that we really don't know the impact of these large expansions on the earliest stages of Huntington's disease. It is still a debate. Certainly we see them in later stages. We don't know about earlier stages. It also suggests that maybe therapies modifying CAG expansion might need to reach these medium spiny neurons that expand specifically. And so a question, which Jong-Min will get into perhaps, is how you might actually modify CAG repeats in these neurons specifically. We also don't know at what stage of Huntington's disease you might need to modify or prevent CAG expansion from a therapeutic point of view. It may be that you need to start it very early to prevent those expansions from occurring in early stages. And then lastly, these loss of interruption variants that dramatically accelerate Huntington's, they don't seem to increase somatic instability, suggesting that there are still other factors that we need to study and appreciate. So with that, I want to wrap up. I think I'm a few minutes over. I want to thank my colleagues at the Hayden Lab, as well as my collaborators at the Hines Lab, at the Rockefeller University, and also our partnership with the University of Auckland, who also has a remarkable brain bank. So with that, I'll hand it over to Jong-Min.
So I thank HDF webinar organized for this chance to share our work. And I also thank Sarah and Chris for nice introduction and covering a lot of groundwork for today's topic. So in my presentation, I'll walk you through how we discovered non-canonical CAG repeat, their significance, and therapeutic strategies. So a little bit of my background. I was born and raised in South Korea. After finishing college and master degrees, I came to US to earn a PhD degree at Madison, Wisconsin. Since then, I moved to Boston to get a postdoctoral training under Dr. Miles McDonald. Since then, I have been committed to HD research, focusing on human genetics. Most of my HD genetics was part of the GeM-HD Consortium, which is an international collaboration to identify genetic modifiers of HD. So in my talk, I'll try to deliver two main messages. Number one, human genetics is informative when developing treatment. Number two, each HD individual is unique and different, and studying differences between individuals may lead to novel treatment strategies. So we focus on human genetics in our HD research because therapeutic strategies that are supported by human genetics can significantly increase the success rate of clinical trials. So far, there are only two genetically supported therapeutic targets in HD. The first one is the expanded CAG repeat, which accounts for all the cases of HD. The other genetically supported therapeutic target in HD are genetic modifiers. Today, I'm going to talk about the therapeutic strategies that target both expanded CAG repeat and genetic modifiers at the same time. So as Chris explained, all cases of HD are due to expanded CAG repeat in the Huntington gene, representing the first therapeutic target that is supported by human genetics. However, no two HD individuals are the same. For example, the sequence of the repeat may be different between HD individuals, Also, HD individuals may have different genetic modifiers. These different flavors and toppings make each HD individual unique, representing the second therapeutic target that is supported by human genetics. These human data show huge variabilities contributed by genetics, highlighting three take-home messages. First, age at onset is inversely correlated with the size of the expanded CAG repeat, meaning longer expanded CAG repeat produce earlier disease manifestation. However, CAG repeat is not the only factor that determines age at onset because age at onset is highly variable at a given CAG repeat size. Lastly, notice these sibling pairs. The sibling pair in green, they developed symptoms 10 years later, while the other sibling pairs in red developed symptoms in 10 years earlier, even though they carry the same CAG repeats. These family data indicate that the variability in age at onset, not explained by CAG repeat, is contributed by genes. Together, observations in HD individuals suggest that while the size of the CAG repeat is important, additional genes may further modify onset significantly. So identifying genetic modifiers is important for developing treatment strategies because they help us understand the mechanisms and inform therapeutic strategies. This cartoon illustrates the concept of genetic modifier and how it can be useful. Imagine two people drinking beer and developing a beer belly problem. Although they are drinking the same amount of beer, Homer on the left has a much larger beer belly compared to Homer on the right. It turns out that Homer on the right managed to keep his beer belly smaller by watching TV less and exercising more compared to Homer on the left. In this example, we learn two key findings, such as mechanism and treatment strategy. First, we can tell Homer Simpson's beer belly problem involves calorie consumption and energy metabolism. Second, Homer Simpson can manage his beer belly problem by targeting the cause and the modifiers. We are taking the same approach that is illustrated in this cartoon to gain insights into HD mechanism and subsequently develop therapeutic strategies. We are taking genome-wide association study approach to identify genetic modifiers of HD. GWAS is a way to figure out Mother Nature's genetic experiment that resulted in different phenotypes of your interest. Mother Nature produced a lot of genetic variations in humans, leading to various disease and non-disease phenotypes. GWAS approaches start from the result and trace back to find out the source that produced the phenotypic differences. In HD, different age at onset corrected with CAG repeat size is a result of some of Mother Nature's genetic experiments. We are taking GWAS approach to search the genomic sequence, aiming to identify genetic variations that can explain earlier or later onset compared to expectation. We performed GWAS based on about 9,000 HD individuals, focusing on age at onset normalized by CAG repeat size. In this genetic analysis, the GeM-HD Consortium identified many significant onset modification signals in the genome, representing mostly DNA repair genes involved in CAG repeat instability. As Homer Simpson learned about the mechanism and treatment strategy for his beer belly problem, these GWAS results are teaching us that CAG repeat instability plays an important role in HD and represent a promising therapeutic target. Surprisingly, GWAS of onset also discovered unexpected association signal at the Huntington gene region on chromosome 4, although Huntington is not thought to be involved in DNA repair. So we investigated this region further, discovering that there are two separate association signals, such as onset delaying signal displayed in green triangles and onset hastening signals marked by red triangles. Follow-up sequencing of HD individuals carrying these signals revealed that there are different types of repeats. For example, more than 90% of HD individuals carry a canonical CAG repeat, which is an expanded CAG repeat, followed by CAA-CAG, CCG-CCA, and CCG repeat. A small number of HD individuals carry expanded CAG repeat without duplicated CAA-CAG, loss of CAA, loss of CCA, or loss of both CAA and CCA, as Chris summarized nicely. Issues arise when determining the size of the repeat by P-cell assay because one of the P-cell primers bind to this region. As a result, the size of repeat with duplicate interruption was overestimated, making that individual seemingly late onset. In contrast, the size of CAG repeat with a loss of interruption was underestimated, making that individual seemingly early onset. So the bottom line here is the significant association signal on chromosome 4 in our initial GWAS were due to incorrect CAG size estimation. Recognizing these issues, the GeM-HD Consortium determined the size of the uninterrupted CAG repeat using a sequence-based method, recalculated age onset, not explained by CAG repeat, and then performed the GWAS analysis again. If you compare this data to the previous result, you can tell most of the association signal remained largely unchanged. The biggest change is the decrease of the chromosome 4 signal, suggesting that the size of uninterrupted repeat is important. Still, the chromosome 4 signal was not completely removed, indicating that non-canonical repeat still produce some onset modifying effects. Our new GWAS result defined by a small number but very precious individuals with a non-canonical repeat taught us two lessons. First, age at onset is determined by the size of the uninterrupted repeat in the DNA, not the size of glutamine in the Huntington protein. The second lesson was non-canonical CAG repeats still produce strong onset modifying effects. For example, our statistical modeling approach showed that having loss of interruption is equivalent to having 3 more CAGs in terms of onset. By contrast, having duplicate interruption is equivalent to having 1 fewer CAG. When thinking about therapeutic strategies, duplicate CAG interruption is directly relevant as it is supported by human genetics and it can delay onset in HD individuals. I mentioned that having duplicate interruption is similar to having 1 fewer CAG unit. On average, duplicate interruption is translated into delay of onset by 3.7 years, which is representing robust onset modifying effects. Then what are the mechanisms by which duplicate CAG interruption delays age at onset? Chris showed convincing evidence that loss of interruption does not alter CAG repeat expansion. In contrast, Zach in our research group showed that CAG repeats with duplicate interruption are less prone to repeat expansion. So this evidence suggests that there are maybe distinct onset modifying mechanism for loss of interruption and duplicate interruption. Based on delayed onset in duplicated interruption, we conceived the therapeutic strategies of generating additional CAA interruption. It is expected that generating additional CAA interruption will target all 2 therapeutic targets supported by human genetics because it is expected to shorten the size of uninterrupted CAG repeat and it will produce additional onset delaying effects. So, what is the maximum onset delaying effects of duplicated interruption? In this example of an HD individual with 43 CAG repeat, the expected age at onset is 48. If the duplicated CAA interruption is generated in this individual early in life, uninterrupted repeat size decreased by 2, and duplicate CAA interruption may produce additional onset delaying effects, resulting in delay of onset by 12 years in total. In some cases, additional CAA interruption may change some of reduced penetrant CAG repeat into non-pathogenic repeats. Potentially, additional CAA interruption may also change some of fully pathogenic repeats into reduced penetrant repeats. So, how can we generate additional CAA interruption? The genome engineering field has developed various gene editing technologies. Base editing is one of newly developed gene editing methods, which can be used to change a specific DNA letter into another, providing a great tool to address disease due to point mutations. Because of this, people think base editing is not an ideal tool because HD is not due to a point mutation. However, we all learned the therapeutic potential of duplicated CAA interruption, and the difference between CAG and CAA is only 1 DNA letter. Therefore, if we employ base editing to convert G to A, we will be able to generate additional CAA interruption, resulting in shortening the CAG repeat and producing additional onset delaying effects. Another benefit of a base editing strategy is that converting G to A does not alter the size or sequence of polyglutamine in the Huntington protein, as both CAG and CAA encode glutamine. To determine the feasibility of base editing strategies to convert CAG to CAA, we conceived 8 different strategies and test them in a cell culture system. Unfortunately, the conversion efficiency of base editing strategy to make duplicated CAA interruption was not high. However, if we target the front end of the repeat, we can convert the second CAG to CAA, fourth CAG to CAA, and sixth CAG to CAA with high efficiency. During my lab also able to convert the CAG in the middle of the repeat by directly targeting the CAG repeat. Since base editing strategy directly targeting the repeat will permanently change the DNA sequence of many other CAG repeat containing genes in the genome, we are pursuing base editing strategy with a relatively high gene specificity, such as approaches to convert the second and fourth CAG into CAA. Focusing on presumably safer base editing strategies, we evaluated the levels of specificity and determined the molecular outcomes. Base editing strategies aimed at converting second CAG and full CAG into CAA target some of the unique sequence in the genome and the repeat region, and therefore they were believed to be specific to Huntington. In support, these base editing strategies did not alter the expression levels of any genes in the genome. Also, as expected, our base editing strategies converted both expanded and unexpanded CAG repeats. Still, we do not predict significant adverse effects of converting CAG to CAA on the normal chromosome because we are not changing the Huntington protein sequence at all. In addition, the total Huntington protein levels were not changed by base editing treatment, meaning that the levels of both mutant and normal Huntington protein will be the same before and after treatment. If the sequence is not changed and the levels are not altered, how this base editing strategy can delay onset? As we saw previously, having duplicate CAG interruption delay onset potentially by decreasing CAG repeat expansion. Similarly, having CAA interruption at the front end of the repeat also decrease repeat expansion in the knock-in mouse model. Taken together, our data suggests that base editing strategies to convert the CAG at the front end of the repeat is feasible, relatively specific, and may delay onset by decreasing CAG repeat expansion. There are many questions to be addressed to understand the mechanisms of non-canonical repeats and to advance to therapeutic development. To name a few, as Chris showed in his presentation, the mechanism of onset hastening effects of loss of interruption still remain unclear. Jaehyun in my lab made a lot of progresses in generating HD knock-in mouse model with the loss of interruption to understand the mechanism of loss of interruption, so we can reverse such a mechanism to slow down HD. In addition, what we can achieve through base editing is different from the duplicated interruption that Mother Nature made. So Jaehyun in my lab also created a knock-in mouse model carrying CAA interruption, and he is looking into whether CAA interruption at the front end of the repeat is safe and produces similar effects as a duplicate interruption. In summary, I shared my view that each HD individual is unique and special with respect to CAG repeat size, sequence of the repeat region, and the genetic modifiers. Studying such uniqueness and differences led us to important insights into disease mechanisms and therapeutic targets that are supported by human genetics. My lab is committed to studying loss of interruption so we can reverse the mechanism to slow down HD. We are also trying to learn from Mother Nature's genetic experiments involving duplicate interruption to develop safe and effective treatment strategies. With all this, I thank the HDF webinar organizer again, the GeM-HD Consortium members and funders. Most importantly, none of our genetic findings wouldn't be possible without participation of individuals with HD, especially a small number of HD individuals carrying non-canonical repeats. Thank you.
Thank you both so much. That was fascinating. And I think I'm going to start by honestly echoing what you just said, Jong-Min, that a lot of this, everything that we just heard today, was only possible because of the participation of the HD community. So people that participate in studies like GeM-HD, which I think it was over 4,000 people, and people that are participating in studies like Enroll-HD that are observational, they are leading to very real findings in HD research that are helping develop treatments and really propelling us forward. So thank you to everyone who participates in these trials. It's so, so important to have the participation of the HD community.
I also wanted to put in a note for people who go through brain donations, which is this ultimate gift to Huntington's disease research. And ultimately, HD is a disease of the brain, right? And so a lot of our most critical questions right now rely on brain tissue that has been very generously donated by people. Yeah.
That's a great point. And I think we're more and more, we're seeing research with human brains, which is so critical, right? Like we can only get so far with animals, and it's very, very important. So great point, Chris. Okay, so thank you to everyone who's been submitting questions. Continue to do that throughout the Q&A. We'll try to get to as many as we can, and I'll kind of ping back and forth between you, Chris, and you, Jong-Min Lee. So the first question is for you, Chris, and the person wants to know if the normal CAG repeat size within the Huntington gene also causes somatic expansion, or is that just the mutant CAG size?
So yeah, I didn't want to get into too many details because this is such a fascinating and broad field. So there is instability that occurs at CAG repeats lower than the HD range. So for example, some of the work that I showed in my presentation was about sperm. So there's instability in sperm well below the HD range. People can have 25 CAG repeats, 27 CAG repeats, and some of their sperm will have more repeats than that. Some of the sperm will have 30 or 31 or so on and so forth. So depending on the cell type and depending on how long the repeat is, yes, there can be instability also in the normal CAG repeat.
Thanks, Chris. So here's one for you, Jong-Min. They're curious about how base editing works. And I know you were talking about the molecular details, but you kind of left out the really kind of the key word in that, which is CRISPR, right? Like that's what a lot of people know from the news stories, but it involves CRISPR, right? So could you talk a little bit about more how base editing works and how maybe CRISPR is involved?
So base editing is composed of multiple components. CRISPR is one of them. And base editing, there are similarities between DNA letter A, C, G, T. There are similarities. Some has methylation group in the base and the other doesn't have. So it is base editing. You just hook the demethylase in the CRISPR system, and then you target the specific DNA letter, demethylate, and then the endogenous DNA repair system try to repair, and then as a result, you can convert to specific DNA letter into another. So that's how base editing is working.
Thanks, Jong-Min. So kind of going back to your point, Chris, about cell type specificity a bit, this person asks, sorry, the question just jumped around. Oh, no, that's not it. Just a second. Okay, so they say it's hard to understand how these long repeats develop in MSNs since MSN should be terminally differentiated and thus not dividing. Can you talk about your hypothesis for this? Or, why do we see this in non-dividing cells?
Yeah, so there are many experts in the field that work specifically on understanding the mechanisms by which expansions occur, because there's actually different ways that expansions can occur based on what's happening with the DNA. And so what's thought to occur in medium spiny neurons is that there's actually two different hypotheses. So one is that there's damage that occurs to DNA just because of environmental factors or because of mistakes that are made in your cell that then have to be repaired by your cell, and that the expansions are a byproduct of that repair of damage that occurs to your DNA all throughout your body all the time normally. Another hypothesis is that it actually occurs because of when DNA is transcribed, that there are slip outs that occur, and that perhaps they are not put back together properly, and then the cell mistakenly repairs it as a longer repeat. So there's actually the biochemistry can get quite daunting, but it doesn't rely on DNA replication. Yes, that is correct.
Thanks, Chris. So Jong-Min, thinking about down the line, right, like getting this induction of CAA interruptions into people, the person asks, for a therapeutic, would generating a duplicated CAA interruption be delivered through gene therapy? How do you envision this being delivered?
So AAV is one way, maybe golden nanoparticle is another way. Potentially there may be a third method, but both of them will be mostly promising method to deliver this base editing system to the brain.
Thanks, Jong-Min. So here's a question, Chris, that talks about something you mentioned about somatic expansion not being present kind of early on in disease with that one patient that was only symptomatic for 1 year. They say, so somatic expansion is not the trigger for HD, as the expansion is not necessarily present in early stage disease. Can you talk a bit about that?
So this is really where the leading edge is on somatic instability research in HD, and we don't really have all the answers yet. So we've seen not only in that individual I showed, but in other early stage individuals that we don't see very many of these. We don't see any of these extra long CAG repeats. However, we are now looking at the medium spiny neurons specifically, which may have a different expansion profile. There's many different kinds of neurons in your brain, right? Medium spiny neurons are just one type. And so the data that I showed was from all brain, a whole chunk of brain tissue, right? But if you look at the specific kinds of neurons that are vulnerable, the expansion profile may be different. And I hope to share with my colleagues as well as the field within the coming months what that looks like. Yeah.
Thanks, Chris. So Jong-Min, this is a question around the GWAS data. They say, how can one reconcile that mismatch repair genes come up in GWAS, but at the same time, you do not see dramatic somatic expansion at age of onset? Would it suggest that high toxicity of protein at approximately 70 CAGs?
So I think the GWAS result is directly relevant because we are looking at humans, and because of that, there are a lot of limitations. We cannot run experiment on humans, but clearly I understand your concern and the conflict, but the human data is gold standard, and we try to learn human data, and I think instability is quite important driver for the HD. So we try to start from there.
Thanks, Jong-Min. Kind of in that same vein, I guess, Chris, this is a person asking about the animal data to support somatic expansion. They say, is there any animal data to support that tackling somatic expansion can delay disease onset? Can you talk a bit about that?
So I would say that there's conflicting data in the field on that point. So without getting into too much depth about the different mouse models that exist in HD, there are some mouse models where if you stop somatic expansion, there's no effect. There's no benefit whatsoever. There's others where there's suggestive benefit. So right now, what we're trying to figure out in the HD field is why some models, when you stop expansion, have no benefit, and others, perhaps there is some benefit. We really don't know why, but clearly there is some point of no return. There's some level of expansion where you can't rescue it anymore. And perhaps the different mouse models will give us some insight to that.
And I guess to add to that, there's a lot of people looking at this, right? Like Vanessa Wheeler's looking at this, Jill Bates, William Yang just put out a paper that's looking at this. And yeah, there's a lot of people interested in this area, but as Chris mentioned, it's kind of conflicting at the moment, but I also don't think it will be a one-to-one, right? Like what we find in mice as these kind of thresholds and these numbers for the CAG repeat, they might not necessarily translate to the exact same in human. And so that kind of goes back to the point that Chris made, why it's so important to study these things in human brains.
Yeah, mice are not people. I say it again and again and again. I mean, we all have different points of view on this, but mice are not people.
So here's one I'll throw to you, Jong-Min. This person asks if they can be involved in research without donating their brain. They have a 42 CAG repeat and they're almost 63 years old at their last enroll visit a few months ago. They were still pre-symptomatic. They've been involved with research for nearly 25 years back in the Pharaohs days, and their neurologist theorizes that they likely have other genetic factors at play.
So yeah, there are ways to be involved in these HD studies. So you are a good example, which may have a good modifiers. Maybe you have a duplicate interruption in the CAG repeat, so you can just supply your DNA samples in the blood, and then your phenotype, lack of phenotype can be a good contributing data to the bigger genetic studies.
Yeah, and I would just add, it seems like you're already involved in Enroll, which that's the best thing you can do.
Yep.
Okay, so here's one for you, Chris. They ask, when not seeing very long repeats in some of the HD patients, do they also see less neuronal death compared with other similar stage HD patients with very long repeats?
So I'm reluctant to tell you an answer one way or the other because we're looking at this right now. So this is, of course, something we very, very much want to know is, do these rates of instability that we observe correlate with neuropathology, with how the brain actually deteriorates in HD? And it turns out that this is not actually a very simple question to answer. You have to look at quite a few brains and have very accurate information about what they look neuropathologically. So all I can say right now is that we are looking at this. Yeah.
Thanks, Chris. So maybe, I mean, either of you could take this, but I think there's a few questions here about CAG repeat size changing over time. And I think this is a concern for people living with the expansion in the HD gene. Do they need to get tested again?
No.
Can you talk a bit about why that is?
The short answer is no. Yeah.
But why is that? Can you talk a bit about that?
So blood is actually among the most stable of tissues. And the cell types you find in blood are among the most stable in terms of the repeat. So even though there is some instability and some small fraction of the cells in blood will change size, what will come up is the main size that you get from a test result. We've never seen that change. Looking at many, many hundreds of people, that's never changed. Yeah.
Thanks, Chris. So Jong-Min, here's one that might be a bit more technical, but they're asking if you can talk a bit more about how to avoid off targets of G to A base editing.
So as I described before, the base editing involves CRISPR, and if you use CRISPR in a way, the guide RNA binds to specific DNA sequence in the genome, maybe you can generally decrease the off targets. So that's why we are focusing on the base editing strategy involving some of the unique sequences in the Huntington gene region.
Thanks. So I think I'll ask, this might be our last question. I'll ask both of you to answer this. And the person says, do you think the pathogenesis is mainly driven by expanded DNA repeats or toxicity caused by Huntington RNA and protein accumulation? Who wants to go first? You want to go first, Chris? You want to tackle that?
Sure. So, I mean, this is the question in the field right now. I mean, whoever has asked this question has really hit the nail on the head. And I think if you ask experts in the field, everyone has sort of a nuanced point of view. I think probably the most common point of view is that expansions are necessary but not sufficient. So you have to have expansions, but that there are other consequences that maybe become no longer dependent on CAG repeats. So maybe there's some expansion that occurs first, but then other mechanisms take over. But there are people on other extremes who think that only very long repeats are pathogenic, and then other people who think that, well, you don't really need somatic expansion. What you inherit is actually maybe even enough. So there's points of view all along that spectrum. But I think most people would say you need some somatic expansion, but it's not sufficient to elicit the disease on its own.
It's a great question. So if you look at the human data, DNA, RNA, or protein, which is important? I think based on human data, what I can say is the age at onset and other phenotype is best explained by DNA, CAG repeat size. But I'm not saying RNA is not important. I'm not saying protein is not important. The human data is best explained by the CAG repeat size in the DNA.
Thanks. Yeah. And I agree with what you said, Chris, right? Like this is the question right now with all of the somatic instability data coming out, like it's kind of flipped everyone on their head and we're reconsidering.
And, I want to just very briefly touch on what I talked about with respect to onset, right? So we know the longer your repeat, the earlier your onset. And we still don't actually know why that's the case. So somatic instability does provide a potential explanation for that, but we don't really know if that's the reason either. But it's compelling. Yeah.
That's a great point. All right, so we are at the end of our time, so I'm going to wrap it up there. If you had a question that we didn't get to, which there are quite a few, I will send those over to our speakers so they can follow up with you if you left your contact information. I want to again end by thanking everyone who donates to the HDF to support the research that we fund, and of course, everyone for tuning in. And last but certainly not least, I want to thank our speakers, Jong-Min Lee, Chris Kay. Thank you so much for taking the time out of your busy schedules to share your very fascinating and incredibly important work. It was very much appreciated.
Thank
Our
you.
next webinar, thanks. Our next webinar is Tuesday, March 25th. We are going to hear from Professor Jill Livingston about her work heading up the Lancet Commission on Dementia Prevention, Intervention, and Care. She's going to share their recent update on what we can all do to keep our brains as healthy as possible and prevent dementia. So definitely tune in. Registration for that webinar is now open, so head over to the HDF website right now so you can get it on your calendar. Thank you all so much for joining, and I will see you next month.